right, hello again. We're back again for the same sequence that we had last time. Because apparently we were too much in advance, and I apologize for that. We were lacking members of the jury, which I'm thrilled to see now. Hello, dear members of the jury. I'm sure it was going to help you to actually see the pitch. Right, so that's what we're going to do now, and we're going to start again from the top. We are still in the health and biotech category. And we're going to listen again to our first contestant because she was brilliant, and we want to hear her again. So that was rehearsal you just saw. So our first contestant in this category is ALSYMO. It's supported by the SAT Paris-Saclay, the University Paris-Saclay, le CEA et l'INRAE. It develops a first-in-class drug candidate against a rare life-threatening disease. And we are now going to listen again to Sylvia Cohen-Kaminsky. Sylvia, you now know the rules because it's the second time. Sorry again for that. The floor is yours for 3 minutes.
Thanks. ALSYMO is committed to fighting pulmonary arterial hypertension, a rare respiratory disease. Imagine feeling out of breath all the time and being restricted in your everyday life. That happens to PAH patients. The cause of PAH is vascular remodeling, leading to high pressure in the blood, and finally, right heart failure and death. There are 14 drugs on a growing 7 billion market shared by 6 major companies, 2 blockbusters, but they are only vasodilators that improve survival, and 1 of 10 patients still dies every year. Thinking out of the box, ALSYMO is developing an anti-remodeling drug. The target, unexpected, is the NMDA receptor known to mediate cognitive function in the brain. And we discovered, 1, that this NMDA receptor is engaged in PAH lung arteries, making it a relevant therapeutic target. 2, our drug candidate is a peripheral receptor NMDA antagonist that do not go to the brain to avoid brain effects. And we are inventors of 2 patents protecting 2 chemical series granted worldwide with acquisition of licenses from SAT Paris-Saclay. 3, we have a proof of concept. Our antagonist inhibits lung vascular remodeling and pressure and improve survival over the standard of care. And finally, our NMDA receptor is a molecular hub at the crossroad of PAH pathways, and this is our competitive advantage because it can target every pathway at once. We are a multidisciplinary team with key opinion leaders from the National Referral Center of PAH, and together we have the ambition to provide the proof of efficacy in humans within 5 years, and we now need 10 million to complete regulatory and phase 1 studies, and then we'll have 2 phase 2, 1 in a subgroup of patients called PVOD. Their outcome is the worst. 2 out of 3 will die within 2 years. Since they do not respond to vasodilators, this is the unique opportunity to test our product as monotherapy in naive patients. And the second trial in classical PAH as add-on to the standard of care. PAH is a very dynamic field in terms of acquisition and M&A activity, and tech transfer to pharma, early or late, is what we want for ALSYMO. Thank you.
Thank you very much, Sylvia, for doing this again for us, and apologies for making you run this pitch a second time. The next startup is a member of the 2022 promotion from Gene.io, an acceleration program from Genopol. It has also collaborated with INSERM and the APHP, the Greater Paris University Hospitals, to create rapid and non-invasive diagnostic tests dedicated to the follow-up of kidney transplants. It's called CGenetix, and it's represented by its CEO, Geoffroy. Geoffroy, the stage is yours for 3 minutes, and the 3 minutes start in 3, 2, 1, go.
Kidney transplantation is the gold standard treatment for end-stage kidney disease. Unfortunately, 1 in 4 individuals face two kidney rejections within 5 years after the surgery. As you may have guessed, kidney transplant is a precious organ that is subject to rigorous monitoring. Current diagnostic methods are based on urine and blood tests to predict kidney transplant rejections, while renal solid biopsy is used to characterize and confirm kidney transplant rejections. However, current diagnostic methods are not very sensitive. They are limited. They are not very sensitive. They are invasive and painful for the patient, and they are very long to set up. At CGenetix, we propose an in vitro diagnosis test based on cell-free DNA analysis. Our patented technologies is able to screen and characterize kidney transplant rejections. We precisely quantify DNA molecules released by kidney into biological fluids such as urine and blood. We already validated a clinical proof of concept of our technologies in 100 transplanted patients. Our minimal viable product will be able to divide it by 5 the number of silent rejections to reduce early graft losses, to reduce by half the use of renal solid biopsy, and also to reduce by 20% the hospital expense related to patient's care. The liquid biopsy market dedicated to the transplantation is a fast and dynamic growing market. The CAGR is estimated at 26%, and the total market is estimated to reach $6 billion in 2027. Main actors are present in this market, such as CareDx, Eurofins, and Natera. Regulatory and reimbursement pathway already exist for our product type in the US market. To reach this market, we have a 5-year roadmap divided into 3 phases: identification studies, pre-validation studies, and registration studies. So these 3 phases will require a total investment of 10 million euros. We expected to reach 100 million euros of annual gross revenues with this product in 2030. Our team is the result of a passion for life science and is composed of business and scientists. We are also mentored by experienced key opinion leaders in nephrology transplantations and also by experienced CEO of in vitro diagnostic company. Finally, we would like to finalize the prototype and to validate it in a prospective studies, we are now looking for 500,000 euros pre-seed round. If you want to join our journey and contribute to save patients' life, kidney transplant patients' life, feel free to contact me. I am Geoffroy, the CEO of CGenetix.
Thank you very much. This is for the next contestant. And our next contestant actually already has 2 products that are active against one of the scourges of the earth, cancer. It's incubated at Incube Alliance. It's called aKen Medical, and it's at the forefront of a novel approach to radiotherapy in collaboration with institutions like the CEA. We now welcome its president, Laila QUERE, who has recently, you were recently awarded the New Technology Venture Accelerator in Los Angeles during the entrepreneurship. That's quite good already. Laila, you have 3 minutes on the stage, and your 3 minutes starts now.
Hi, I'm Laila. I'm the CEO of aKen that proposed a new cancer therapy based on alpha radiation and nanomedicine. Many cancer treatments are non-selective. They lead to side effects. Many cancer patients have to choose between extending their life and the quality of life. So clearly, we need a change, and we need to do better. In this context, aKen Medical proposed its new cancer therapy based on its technology, DNAR. So DNAR, it is a carrier that contains the emitters of alpha radiation, which is a very powerful radiation. DNAR also avoids the release of emitters in the healthy tissues. DNAR, it is injected into the tumor directly as a radiopharmaceutical preparation. It does not need any external instrument to be activated, and DNAR keeps radiation only in the tumor, improving targeting and sparing healthy tissues. DNAR can also be sealed on a device adapted to the shape and location of shallow tumor. For example, for skin cancer, the device will be a bandage. and it is designed to deliver the radiation only the tumor, sparing healthy tissues. So since our technology is global and can be applied to all tumors, so our market is the cancer therapeutic. But we have selected as the first application the pancreatic and skin cancer. Today, several companies and a lot of funding are ongoing. But aKen is unique by proposing a single technology for brachytherapy and pharmaceutical approach involving nanomedicine that improve effectiveness and reduce side effects. aKen has a strong team with many experience in nanomedicine, chemistry, radiology, and business. We have also the privilege of having an amazing board of experts. We have also developed key partners to develop our technologies. Today, our first results on mice are very promising with effectiveness and low toxicity. We are looking for 2 million seed funding to build out our team and for more preclinical tests. And our future goal is to set up the first clinical trial in 2027, and we will look for partners for our go-to-market strategies. Thank you for your attention. I would be happy to connect if you want to learn more about aKen, and I would love to take your questions. Thank you.
Thank you very much for the precisely 3 minutes. Thank you very much. We all know that what doesn't kill us makes us stronger. And if you apply this very literally, then you get one of the foundations of our next startup. It's called V4Cure, and it develops innovative drugs derived from powerful molecules present in animal venoms. It's a spin-off from the CEA, and it's represented by its CEO, Sonia ESCAICH. Sonia, the floor is yours. 3 minutes. And your 3 minutes will start in 3, 2, 1, you're up.
Hello, I'm here today to look for investors in V4Cure. So why I'm showing you this pretty scary green mamba? Because animal venoms contain highly potent biological molecules, and V4Cure mission is to develop innovative drugs derived from animal venoms. But don't worry, we don't raise this animal in the lab. The company was created recently as a spin-off of CEA to develop innovative drugs to treat cardiorenal pathologies. Having spent many years in pharma R&D and as a biotech entrepreneur, I'm very pleased to be part of this great team because of the massive potential of this new molecule to make effective drugs. Our science is supported by key opinion leaders in the field, and that's because our first asset, peptide 232, answers a medical need. Peptide 232 is a solution to treat hyponatremia. Hyponatremia is a pathology where low level of sodium in the blood have dramatic effects on millions of patients today. This pathology has no effective treatment, and 2/3 of patients go untreated. The market potential is high, with a competitor drug sale of $0.7 billion US, and this by treating less than 10% of cases, sorry. So there is room for V4Cure drug on these markets. We have very well validated proof of concept data showing that our peptide 232 is better in terms of safety and efficacy than current treatments. Our business model is based on partnering with pharma for our go-to-market moving. And we have a clear development path to achieve clinical proof of concept in clinical efficacy in phase 2 and creating value by 2027. Our aim is an exit via an M&A or IPO. To succeed, we will need a Series A funding of 8 million to go to IND and phase 1. in phase 1, we will have a diuresis biomarker that will allow to have a first proof of efficacy. And the next step will be a Series B of 10 million to achieve phase 2 clinical trial. Today, we are looking for seed funding. And I will be happy to meet you afterwards. Thank you.
Merci beaucoup. Thank you very much. And it's now time for our final pitch of this 2023 Spring 50 contest. And our very last contestant is acting in a very exciting and promising field. It's spearheading the development of a new and highly efficient process for producing messenger RNA. It's called ByoRNA. It was founded by Pascal VIGUIÉ, who has a Master of Science in Mathematics and Physics from l'École Polytechnique. Pascal, the floor is yours for the last 3 minutes of the day.
Thank you very much, Alexandre. Hello, everybody. Thank you very much for being here today. I'm going to present you ByoRNA. Our goal is to produce long messenger RNA for new RNA therapies. We believe that the fate of RNA therapies will be similar to what happened to biologics during the last 20 years, starting from a very small part of the drug market. Currently, the biologics is a $300 billion market, and we are confident that the RNA therapies, which application to vaccines, as we have seen during the pandemic, but also to oncological cures, and application for protein replacement will be a huge part, at least 20% of the future drug market. So what problem do we want to solve, and what are going to be our main advantages? First, our technology is going to replace, that's our aim, the current in vitro production. And the problem of the current production is that it's very expensive. 1 kilogram of RNA costs around 50 million euros. And it's limited in volumes. And it only produces shorter messenger RNA. So we want to divide the prices, but at least 50, and to increase the volume and the length. Just before talking of our competitors, the way we're going to do it is by creating a small intelligent safe inside the cell, which recognizes the RNA and which protects it. So there are a few emerging players. Of course, Moderna and BioNTech are already there, but with a technology we cannot be efficient at high volume, and which is very expensive. And our main competitor is an American-based company, Sensible Biotechnologies, which has just raised $4 million, and which partners with Ginkgo Bioworks. We plan to raise now 1 million in pre-seed, half of it in dilutive and half in non-dilutive. And we want to do a seed round next year of 7 million in order to be able to have, within 2 years, our first production capacities. So we have been formed by Thierry, who is a veteran from the pharmaceutical industry, with 30 years at CGenetix, Merck, Sanofi, and myself, a former entrepreneur. And we are looking for 1 million euro, half of it in non-dilutive and half of it in dilutive. And we want to use this money to multiply by 3 our yield. Thank you very much.